Hello all, welcome back to the BioRocks channel. In this video today, we are going to learn about the overview of the C3 pathway that is going to be your light independent reactions or what we call as dark reactions. Okay, so if you remember, I just told you in one of the videos that the assimilatory power that is going to be the ATP and the NADPH2s which have been produced during the light reactions are being used, are utilized for the formation of the glucoses in the C3 pathway, that is nothing but light independent reactions. So our topic for this video is going to be independent, light independent reactions, which are otherwise called as dark reactions. Okay, dark reactions. This was being put forward, discovered by Mr. Calvin, okay, and Melvin Calvin, and he was being assisted by Mr. Benson. So we call this cycle even as Calvin-Benson cycle. Calvin-Benson cycle. Okay, so he was a renowned plant physiologist in the year of 1961. He has actually received a Nobel Prize. If you remember, it was chemiosmotic hypothesis, which was being put forward by Mr. Peter Mitchell in the year of 1961, but received the Nobel Prize in the year of 1976. This was the year where Calvin has discovered and has got this particular Nobel Prize for these discovery of the carbon pathway in the plants, that is about the fixation of the carbon dioxide as carbon and how the glucoses are being formed. And this cycle is being called as light independent because it is nevertheless not depending on the sunlight for the energizing process, but instead what it does is, if you remember, there were light reactions, children, there were light reactions actually which were dependent on the sunlight, the photons, okay, the discrete packets which were comprising of the energy called as quanta. And we could see that the two end products of the light reactions were the ATP as well as the other one which was being called as NADPH2. These two were being called as the assimilatory power, which were being utilized. They are being utilized in the dark reactions for production of, okay, they are used in the dark reactions for production of glucose, for the production of glucose during photosynthesis process. This is going to be the dark reactions. If you remember, I told you once that it is going to be the 6 carbon dioxide molecules, the 12 NADPH2 molecules, and 18 ATP molecules are going to participate in the dark reactions, which results in the formation of one glucose molecule where the 12 NADPs, okay, the oxidized ones are being produced, as well as you can see that it is going to be the 12 hydrogens which are being released, and the ATPs, you can see that they are going to be 18 ADPs with the 18 inorganic phosphate molecules which have been released, because we are using up the ATP as well as NADPH2s. So this is going to be oxidized and this is going to be broken down into ADP and the inorganic phosphate molecule, and the energy released will be utilized for this particular formation of the glucose molecule. So first, coming on to children, when I speak about this particular cycle, I have to speak about the one of the sugars. This particular cycle is running in the where is the site is going to be? The site is going to be in the stroma. Okay, okay. This is going to be the stroma of chloroplast. Stroma of chloroplast. Where were the light reactions running? In the thylakoid membranes, right? It was in the thylakoids or the discs, what we were calling as. This is going to be the site for the light reactions. Whereas the dark reaction, site of action of dark reactions is going to be your stroma of the chloroplast. Okay, so we are going to start the procedure now. So as we are coming to this particular thing, I told you that it's going to be the carbon dioxide, which is going to be fixed through stomata. So these are entering through the stomata. So it takes up the carbon dioxide, and the carbon which is being present, let me take a different pattern of this. So the carbon which is being present here is going to be fixed. Okay, it's going to be fixed with what is going to be one of the, what you call is, five carbon atom compound already present, which is a sugar, which is being called as ribulose biphosphate or bisphosphate. We are going to call it as ribulose bisphosphate or biphosphate is going to be the sugar which is being present, which is a ready-made sugar already present inside the stroma of the chloroplast. So as once the carbon dioxide is going to be entering into the plant, that is going to be entering into your, as diffused and enters into this particular stroma. Now what happens is the CO2, the carbon which is being present, is going to be fixed with this five carbon atom sugar, and we are going to get a formation of a sugar which is being called as a hexose sugar, which has six carbon atom compound. But this is going to be very, very unstable. The moment it gets formed, it is going to be what you call is, it is going to instantly split into two molecules, okay, and these two molecules each are going to contain three carbon atoms, and this is being called as, we are going to call this particular thing, the three carbon atom compound, as a phosphoglyceric acid. This is being called as phosphoglyceric acid, or you can even call it as phosphoglycerate. Okay, it can be called as phosphoglycerate. And in many of the IB international curriculums, they have used it as glycerate 3 phosphate. Glycerate 3 phosphate, you can use any of the terminologies, but remember that they have only three carbon atom. Okay, so it's going to be one of the carbon atoms is fixed to this sugar present in the stroma, and it forms an unstable compound which splits into half, that is into the two molecules each containing three carbon atom compounds, that is going to be phosphoglyceric acid. Okay, it's going to be this, the three carbon atoms which is going to have a one of the phosphate molecules. So this is going to have a phosphate molecule here. This is going to be the PO4 molecule. Okay. Now coming on to, so we could see that it is going to be the one, it's going to be, we saw that the carbon dioxide, the carbon fixed to the ribulose biphosphate forming hexose sugar. It split into a three carbon atom compounds called as phosphate, that is a 3-glycerate, phosphate 3-glycerate. Yeah, you can call it as a glycerate 3-phosphate, glycerate 3-phosphate, or we call it as what? We can even call it as phosphoglyceric acid. Phosphoglyceric acid. Okay, this is comprising of three carbon atoms. It has how many? Three of them are being formed. It is going to be two molecules. So this is also phosphoglyceric acid, three carbon atom compound from the hexose sugar, which is an unstable sugar. So now what happens is, children, they are going to actually, now, there is going to be one of the ATP molecules which is being consumed here. That is, your ATP is broken down into ADP and the inorganic phosphate molecule, and the energy is utilized here. And that energy which is utilized here is being used in formation of 1,3 by phosphoglyceric acid or di. You can even use diphospho. Many of the texts have used a phosphoglyceric acid here, glyceric acid. Okay, so now coming on to, this is also three carbon atom compound. This was also a three carbon atom compound, but was comprising of what? A phosphate molecule which was attached to the third carbon atom. So we call it as phosphoglyceric acid, or we call it as glycerate 3-phosphate, because this is going to have a phosphate molecule linked to the third carbon. But now as you consumed another inorganic phosphate molecule, that is, it's nothing but as you have consumed up the energy over here, now what happens is the inorganic phosphate molecule is going to get fixed here. So where is this getting fixed is, it's going to be the three carbon atoms. One is already present towards this, but now what happens is the other is being fixed to the first carbon atom. Now this is going to be the first carbon atom, this is going to be the third carbon atom. So we call this as 1,3 di or we call it as bi-PGA, biphosphoglyceric acid, having two of the phosphates on the either sides. Okay, now what happens is the 1,3-BPGA, now in the series of steps, what happens is it is going to lose, it is going to lose is nothing but it's going to lose a phosphate molecule. Now it is going to lose a phosphate molecule, and what happens is it is going to take up the energy from a reduced coenzyme like NADPH2. Okay, and since it is going to add up hydrogens here, so it's going to be oxidized, right? It is going to be oxidized as NADP plus, whereas the hydrogen atoms are being taken up by the 1,3-BPGA. So now what happens is it is getting reduced into a form which is being called as phosphoglyceraldehyde. It's forming phosphoglyceraldehyde. Okay, we can even call this as glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. We call it as GAP molecules, molecules. Okay, this is what is nothing but when it lost one of the phosphate molecule, it was this particular thing which was being lost. So it has a phosphate towards a third carbon atom, but it is getting reduced by this particular NADPH2, which was being formed in the light reactions. Here also, this is from the light reactions has come over here. Okay, and this is the final product which is being produced during the what we call is light independent reactions, which is going to actually form the glucose molecules. Okay, let me come here and give you one simple cycle over here. So the first thing is the carbon dioxide with the ribulose biphosphate, which is being called as RUBP, a 5-carbon sugar present, which forms up a unstable sugar, which was being called as hexo sugar, which was a 6-carbon atom compound, which was highly unstable. And this has formed what it has been split into two molecules of phosphoglyceric acid, which was 3-carbon atom compound. So this was a PGA with 3-carbon atom compound. Now this has consumed the ATP from this particular light reactions, which resulted in the 1,3-BPGA because of two phosphate molecules. We're calling it as diphosphoglyceric acid. And now coming on to here, 1,3 is going to be the phosphates linked to the first carbon and the third carbon atom. And next what happens, it is going to be consuming the next assimilatory power, that is a reduced coenzyme from the light reactions, which is going to result in the reduction process, and this is going to result in the formation of phosphoglyceraldehyde. Okay, this is also going to be a 3-carbon atom compound, but there are specific set of names given to these reactions where the carbon dioxide is being fixed to the RUBP. This is being called as a carbon fixation. This process is being called as carbon fixation, and where the NADPH2 is being used for reducing 1,3-BPGA, this is being called as reduction process or reducing process. And remember children, this 1,3-BPGA converted into PGAL, what happens is this is going to regenerate, it is going to use for what we call is regenerate what regenerate the RUBP molecules, because if you just go on using the RUBP sugars from the stroma, there should be no shortage of the RUBP, so we need to regenerate. So imagine how many of the RUBP contains how many carbon atoms. It is going to be 5 carbon atoms. So how many PGALs can you utilize for making them as a what you call is making them RUBP is nothing but it's going to be the 10 of the PGALs can give you 30 carbon atom wala. So here you can make it equalize by 6 into 5 carbon atom. That is nothing but 30 again. So 10 of the PGAL molecules are going to regenerate the 6 molecules of RUBP. This is important for our examination point of view. Okay. Another thing, children, here, what I need to specify is the first thing is going to be carbon fixation. The second is going to be reduction happening in the dark reactions. The second thing is, the third thing is going to be the process which is being called as regeneration of these RUBP molecules which are happening. Okay, these are the three things that you need to understand. The next thing that is happening here is Your PGAL molecules which are being present here are utilized in the formation of sugars here, which is nothing but your glucose molecule. So what can you call this process as? This process is being called as synthesis process. The fourth thing that is happening here is going to be the synthesis. But many of these, what we call is international boards have specified the carbon fixation, the reduction, as well as regeneration as the three processes happening in the dark reaction. But remember that at last your PGAL, which is a three carbon atom compound formed, is going to be utilized in the formation of the glucoses. Okay, and these glucoses can also again form up other form of carbohydrates like starch. Okay, and they can even form celluloses. Okay, and they can even form sucroses, which we can see in the process which is being called as a gluconeogenesis, where glucose is being utilized in formation of the other sugar molecules. Okay, and coming on to another thing, children, what the PGAL is used, what is the fate of the PGAL, the terminal thing which is being formed. This is your product of your what we call is dark reactions, that is going to be PGAL, which is a phosphoglycerol here, is going to be, firstly it is going to be used in the formation of the glucoses. Okay, and it will be used in the gluconeogenesis process, where it the glucose can be utilized in the formation of many other forms of carbohydrates. The phosphoglycerol, which is a glycerol 3 phosphate, will also enter into the glycolysis process. If you see the biochemistry of the glycolysis, you see that there is formation of phosphoglycerol, which will be converted into one of its isomer, which is being called as dihydroxyacetone phosphate. If you remember, children, this is being called as dihydroxyacetone phosphate. Okay, so DAP, it's an isomer, so they are interconvertible. These two together are forming the glucoses. Okay, so you can see PGAL participating even in the glycolysis. So from here it can even just go into the glycolysis pathway, or it can form the glucoses. Okay, this is a fate. Okay, this is a product, and we are speaking about the fate of the product which is being formed here in the dark reactions. And the next thing is a PGAL can even be converted into acetyl coenzyme A in the cellular respiration process. And this acetyl coenzyme A will be utilized in the formation of fatty acids. It's being used in the formation of your fatty acids. These fatty acids will be making up the phospholipids, which are utilized in the formation of membranes, cell membranes, and the organelle membranes, and also they will be utilized there. So even fats. And imagine, children, if the glucoses, you all know that glucoses are going to have the carbon, hydrogen, oxygen. So imagine that if you just add up these particular carbon, hydrogen, oxygens with a nitrogen from a soil by the nitrogen fixation process, can't you make up amino acids? So this is a way your PGAL is contributing in the formation of your what you call is amino acids also. And again, the sugars having the carbon, hydrogen, oxygen, if you just add up the phosphates here, if you are adding phosphates, can't you make out your nucleic acids? Yes, you can synthesize your nucleic acids also. So imagine that these are all the things. So PGAL may enter into the formation of the glucoses, it may enter into the glycolysis directly, can form the fatty acids, and if we are combining it with the nitrogen, it forms amino acids, and with the phosphates, it's giving you nucleic acids. So this is a wide range of products that can be produced from the PGAL. Okay, so this is very, very important to remember, children. So what are the reactions that are happening in the carbon, that is your dark reactions, is going to be the carbon fixation, the reduction process. We are using the assimilatory power. We are using the assimilatory power which is being produced from the light reactions in fixing the carbon and producing your glucoses which are being present. And what is the isomer for your PGAL here? It's going to be DAP, the dihydroxyacetone phosphate. So imagine that since the first stable compound formed here, What is a stable compound? This was unstable compound. The first stable compound formed here is a 3-carbon atom compound. So we call our dark reactions as, there is another name for this. So we are going to call our dark reactions as C3 pathway. And this was being given by Calvin, calling this particular cycle as a C3 pathway here. Okay. So the C3 pathway, what is it trying to do is utilize these energies from your produced from the light reactions and generating a 3-carbon atom compound, which is going to be otherwise used in the formation of your fats, amino acids, or we enter into glycolysis. There are wide ranges of functionalities of the PGAL, and it is otherwise going to synthesize your glucose. So that is being called as a synthesis. But remember that there is a specific enzyme which is responsible for the carbon fixation here and making up a hexo sugar, that is being called as rubisco, which is being called as a ribulose biphosphate enzyme, which is going to be a carboxylase. We call it as a carboxylase. And what is RUBP? Ribulose bisphosphate. Okay, so the same compound enzyme is going to be the rubisco, which is RUBP's carboxylase. You can even call it as RUBP carboxylase. We can call it as carboxylase. And there are many enzyme contributions in this particular cycle. Okay, enzymes as well as even the minerals are participating, which are nothing but your coenzymes. Okay, coenzymes are also participating. Okay, so even the magnesium ions and even the hexases, there are many other sort of like your enzymes which are participating in this particular reaction. So remember, this is going to be the thing about the C3 pathway. So you can even call this as, see, we have seen in the first stages that we can call these as with several names of light reactions, dark reactions, or we can even call it as a C3 pathway. C3 pathway. So this is a way how your ATPs and NADPH2s are utilized in the formation of glucoses. Okay, I hope children you have understood this video. If you like this video, please hit the like button. Okay, and please share and subscribe my channel. It's going to help you in building up the concepts for your examination point of view. Okay, thank you all.